Hello everyone, welcome to the BIA's webinar on advances in research and development in rare diseases. It's Hannah Johnson from the BIA here, and I'm delighted that we've got four BIA members speaking today. We've got Ben Edwards, Chief Executive Officer of Summit Therapeutics, David Connors, Chief Executive Officer of Nightstar, Gideon and David Speller of Helix, and Alistair McKinnon of Chief Medical Officer of Mereo Biopharma. Just a few things before we start. If you aren't speaking, if you could mute yourself just to stop background noise. This just creates a better noise for everyone else on the webinar. And we also will be recording the webinar, so please keep looking out for an email from me in your inbox with the recording and the slides post-webinar. We will be doing questions at the end, so if you think of any questions throughout, either hold it until we unmute everyone at the end, or there is a chat function in the right-hand corner, so you can post your questions throughout and I can read them out at the end. So first up, I'd just like to introduce Ben Edwards of Summit Therapeutics, who will be speaking about their company and what they're doing in rare diseases. Over to you, Glenn.
Well, thank you all very much. Here we go. I can't see the screen, the slides at the moment.
Have you got the arrows? If you move the arrows, you'll be able to get to your slides.
All I've got on the slides were there, and now all I've got is the BIA Webinar Advances in Research and Development in the meeting number.
Okay, let me just do something 1 second.
Okay, why don't you do the slides and I'll say next slide, please. That's probably the easiest way. Okay, so first of all, as we're, next slide please, we're a public company. I just need to alert you to the fact that my ability to predict the future is worse than all of yours, and so when I'm talking about the past, it'll be accurate, but my predictions of the future will be not that great. So next slide, please. So Summit's really interesting from this webinar point of view because we've got two dramatically different programs. The main thrust of the company and most of the investment money is going into our program on Duchenne muscular dystrophy, where we are using an approach that came out of the team at Oxford University that's called eutrophin modulation. And one of the huge advantages of this approach is that it can treat the entire Duchenne population, irrespective of where the fault is on the dystrophin gene that causes the disease. And we are at the start of a phase 2 program. And obviously, with this leading science, we have to maintain a deep collaboration with the genetics group at the University of Oxford, and that's one of the characteristics I'm going to talk a little bit more about. But in complete contrast to that, and it's actually quite useful, we have a novel antibiotic for the treatment of C. difficile infection, and another quite hot area, you would think, from reading the newspapers and watching the TV. So C. difficile is one of the top 3 problematic bacterial infections. So CDC here in the US and in Europe, it's highlighted as one of the greatest needs in terms of new treatments for this bacterial infection. And furthermore, we've got an antibiotic which is totally new in its chemical space. It's from a new class of antibiotics. And not only does it treat the initial infection very well, but one of the big bugbears of this disease is that it keeps coming back. So you've got about a 25% chance of getting it again within 30 days if you've had it for the first time. We've shown very strong data from a phase 2 study with statistical superiority. And one of the hot areas of antibiotic research is to try and get highly selective antibiotics because you don't want to do much collateral damage. And there are about 1 million cases a year of C. difficile infection in North America and Europe. And you'd think that that would be the bigger market opportunity and that investors would be rushing into it. But actually, it's the other way around. So we have the most interest and probably the biggest commercial opportunity from our DMD program, which has the potential to treat maybe 50,000 patients in the developed world, as opposed to the new antibiotic, which causes, which is for a disease that affects 1 million people a year and causes quite significant death and morbidity around that. So it's an interesting dilemma we've got. And just in terms of weight of money, we've had great difficulty in the past raising money for our antibiotic, and we've only got it to this stage because of the Wellcome Trust, who stepped in and funded the preclinical. They funded the entire phase 1 and most of the phase 2, whereas investors have been really interested in our Duchenne program. Next slide, please. So just a little bit about Duchenne to put you in the picture. It's a fatal muscle wasting disorder. So boys are diagnosed around 3 to 5 because of their slower motor development, and then their muscles progressively waste away. So while they're up to about 10, you've got the rate of new muscle deposition outruns the rate at which the muscle fibers are wasting away, and so they grow and put on weight and gain in abilities, but then they start to decline. They'll be in wheelchairs in their early teens, and with great supportive care now, many of these boys will die in their early 30s, but it's a terrible a long drawn out process. It's interesting in that it's one of the most common fatal genetic disorders, and there's no racial differences at all. It's just a uniform incidence rate across the world by both geography and race. It means there are about 250,000 patients worldwide, and 50,000 of those are in what we would call the developed world. And what's happened is that there's a structural protein in muscle called dystrophin. The gene for dystrophin is on the X chromosome, and so if you have a fault in that in your male, you don't have an additional copy of the gene, and that's what leads to the disease. There's very few really unlucky females who happen to have 2 different mutations on the 2 genes, and they get the disease as well. But effectively, it's a disease of males, and it's a huge protein, and the gene is even larger than you would predict as a lot of introns and exons, and so actually a third of cases it's just a spontaneous mutation, so it's not being passed down from the mother, it's just a mutation of that particular gamete. Next slide, please. So what we're trying to do is something that's really quite interesting. The protein dystrophin links the contractile actin from the muscle fiber to the cell wall and acts as a kind of shock absorber. And so it's sort of arranged in rings around the muscle fiber. But it turns out there's an almost identical form of the protein. I hesitate to call fetal form, but it is expressed in when newly formed muscle fibers are laid down or during repair. So you or I have the ability to make utrophin. It's just that it's usually turned off unless we've over-exercised ourselves in the gym. So at the top of this slide, what you see is in a normal muscle fiber, when it's first laid down, utrophin will be expressed. These are the red dots. But then as the fiber matures, the utrophin will be turned off and dystrophin will be called until all the sites in a mature muscle are occupied by dystrophin. In the middle sequence is what happens in the DMD boy. So the muscle fiber is first laid down with utrophin, then utrophin is downregulated as the fiber matures and dystrophin is called, but the fault on the gene means no dystrophin. And so the sites become slowly unoccupied. And then there's no shock absorbing capacity at that part of the muscle fiber. And during some exercise or stress, there'll be a catastrophic failure, and that will do two things. The first thing is it will cause inflammation and infiltration, and that actually is what leads to the disease of fibrosis and ultimate muscle wasting. But in the short term, there's a repair, and that kicks into utrophin production. And so in DMD boys, their fibers are constantly cycling around, lots of utrophin, not much utrophin, no utrophin, disaster, lots of utrophin, and so on. What we're trying to do is use small molecule drugs that will modulate the utrophin promoter so that it doesn't get turned off. It continues to produce utrophin during the whole life of the muscle fiber, so that even in the mature fiber, we have utrophin there. And that's proved to be very, very successful in animal models in that it completely rescued the phenotype. Really, we've been challenged to get this through into DMD boys as fast as possible. And I think this is what kind of illustrates some of the huge opportunities in orphan drugs and also the challenges. Next slide, please. So one of the huge opportunities in rare diseases is the weight of influence and in some cases money of the patient groups, and with Duchenne being a relatively large orphan indication, and it being a disease of children, so the parents are heavily involved. There are a host of really activist patient groups that are involved, and they are obviously desperate to try and help with research, to try and help with drug development, to try and help with patient recruitment, to try and influence regulators. So one of the other companies involved in Duchenne is a US company called Sarepta, and they have a drug being reviewed by the FDA at the moment, and there was an advisory panel on to look at the data on this drug earlier in the year, and it had the biggest turnout of people at any FDA advisory panel to date. There were 1,000 people at this meeting. They had to move it to a big hotel. They had to have 2 major conference rooms. And from our point of view, there have been stages of this development where capital market investment has dried up, and the project was almost completely funded for a 1-year period by a group of US charities that are shown on here that provided financial support to get us through various points. And then with the data we generated from that, the company was able then to attract investment. So I think in this field, or in these orphan indications, often the patient groups can be really influential. And just as a contrast, there was a C. diff treatment in front of an FDA panel, so produced by Merck, and they had a tiny room with about 8 people in it. So it's huge contrast of patient interest in these things. But I guess the one thing that I would say has dramatically, in my time in the industry, changed and brought these orphan indications forward is not so much being the Orphan Drug Act and the things that can help orphan drugs from a regulatory point of view, although they are very helpful. But I think the one thing that really has made a huge difference to make orphan indications so attractive from an investment point of view and allowed us to deliver fantastic medicines to these patients has been pricing. And that is also the main problem in antibiotics. So there's been a couple of treatments getting close to regulatory approval, and one has got a conditional approval in Europe for Duchenne. That's the PTC Translarna or Ataloran, and that is priced at around $300,000 per year per patient. And the cystic fibrosis treatments, the various other orphan indications the pricing is can be of this order. And I would say that's the one thing that has made such a huge difference over the last 15 to 20 years to encourage companies like ours to develop treatments. And I think structurally it's one of the things we have to think about in the future, because the number of orphan drugs coming through means that these pricing may not be sustainable in the long term, and trying to strike this balance between enough reward for companies to develop these things with a price that society can afford is going to be really important. And just to contrast that, so pricing for a new antibiotic, is much less. You can be talking about $10,000 if you're lucky, but typically many of these antibiotics are selling for $1,000. And one of the C. diff treatments we're competing with, metronidazole, is $10 for a course of treatment. So the huge difference in pricing is in my view what's really driven the orphan drug indication, and what in the future we've got to ensure that from society's point of view there is a balance between the pressure to control costs but also the incentive to develop treatments here. And I think there is one more slide, but I'll just end there because I want to make sure there's enough time for questions.
Thank you very much, Glenn. As I said, if anyone's got any questions, please do put it on the chat throughout, and I'll read it out at the end. So next up, I'd like to welcome David Fellows, who's our next speaker. Over to you, David.
Thank you very much. Can you advance the slides for me as well?
Yes, I can. 1 second.
So just real quickly, go to the next slide. Overview of Nightstar. We are an ophthalmic ocular gene therapy company, and we're exclusively looking at treatments for a range of inherited retinal diseases. Our initial funding came from Syncona, who's the venture capital arm of the Wellcome Trust, and then more recently the Series B was funded in November last year by NEA, which is a blue chip investment fund in the US. The work comes out of Oxford University from Professor McLaren, who's a retinal specialist up at Oxford, and he actually started the work back in 2011 dosing patients. The lead program is for Choroideremia, which is an inherited retinal disease where we introduce a functional copy of Rep1, which is the either mutated protein or null protein missing for these particular patients. So you can go to the next slide. Just briefly on choroideremia, there really is no treatment today, and it does lead to blindness. So patients are born with it, and literally by their 4th or 5th decade, they are functionally blind. Much like Duchenne's, it's obviously an orphan disease, and it's also X-linked. We think the prevalence is somewhere around 1 in 50,000. So if you do that math, there are probably 12,000 to 13,000 patients between Europe and the US that have choroideremia. As I said, it's X-linked, so it affects almost exclusively males. And what happens in this particular disease is patients have this mutated protein. This protein is Rep1, which is an escort protein. So it's required to traffic product out of the photoreceptors to the retinal pigmented epithelium in the retina. And this regulation allows the photoreceptors to get rid of waste product, which is a function of their metabolism, and then that waste product is taken up in the RPE, the retinal pigment epithelium, and phagocytosed. If you don't have this protein, you can't eliminate this waste product in the retina, and ultimately it causes a degeneration in the retinal pigment epithelium, which is supporting the photoreceptors. And of course, the photoreceptors are responsible for converting light signals to electronic signals that ultimately go to the brain. So you can imagine if your photoreceptors begin to die, you ultimately will go blind, and that is indeed exactly what happens in choroideremia. The way that this disease presents is patients usually go in their early teens, and they complain of night blindness. So it affects their night blindness early on, and then it moves to start to affect their peripheral vision. And we all have about 140 degrees or 150 degrees of peripheral vision. These patients slowly lose that, where their peripheral vision starts to close in on them until ultimately they're looking down a very narrow tunnel, almost like looking through a straw. And that usually is by the time they're in their 40s. And then it starts to cross across the central vision, which is what we refer to as macular vision. The macula is the area that's responsible in the back of the eye for your detailed central vision. Once it crosses into that area, of course, you ultimately lose your near vision, your detailed vision, and essentially you are blind. So there really is there's no hope for these patients once they're diagnosed. At a minimum, they're just able to function up until the point where really they can no longer operate out in the public, they can no longer drive. Of course, ultimately they cannot work as well, so they become a real burden on society. So we have a very good economic case, certainly, for treating these particular patients. Next slide. So very quickly, our history actually starts back in the early 2005 period, where we had a mother whose son was diagnosed, named Tommy Salisbury, with Choroideremia, and as a result of that, she decided she wanted to find a way that her son could be treated for Choroideremia before he ultimately went blind. So she began a very aggressive grassroots campaign to raise some money. She was very successful in doing that, able to put together $300,000 that went initially into Imperial College, and Imperial College did the characterization of the Rep1 protein, and as a result of that, we were able to, or I should say, the Wellcome Trust was able to fund the initial work up at Oxford University by Robert McLaren to dose the first patient using this gene therapy. And that, that trial has continued up until this year, last year, where Robert has now dosed 14 patients. And I was able to publish some long-term data, which I can share on the very first 6 patients, which I'll share in another slide. In addition to that, in March of last year, we were able to get orphan drug designation and move forward in a program in the U.S. And we also had 4 investigator sites, one in Alberta, Canada, one at Bascom Palmer in Miami, one in Tübingen, Germany, and also at Oxford. So we've had 4 investigator sites dosing the AAV2 Rep1 gene for these patients. And concurrent with that, we were able to get some additional funding back in November, as I said, last year. And we also have an IND approved, which occurred in March of this year. So we're set to really begin, we'll have a discussion with the FDA on our end of phase 2 data, and then move to a pivotal trial within the next 6 to 12 months. Next slide. From just a highlight standpoint, as I said, we've now completed dosing on 32 patients with some promising efficacy signals and no safety events. As I said, the IND has been approved, and we've got some long-term data out to 44 months on these patients. So one of the big question marks around gene therapy is it durable? And certainly in the patients that we've followed the longest, we've seen continued expression and durability, which is very encouraging. And in addition to Kroyterenia, which is our initial program, we've got 2 other proof of concept studies in 2 other inherited retinal diseases that we intend to start in 2017, which I can share briefly with you in another slide. And then we have 5 other programs that we lifted out of Oxford University through a license agreement. So we've been working very collaboratively with Oxford University and Robert McLaren in the inherited retinal disease space. Next slide. This is the long-term data on the 6 patients, and what you see here, the scale is visual acuity, which is the standard measurement for improvement in one's ability to read a standard Snellen chart or ETDRS chart. essentially the eye chart that you read when you go into an ophthalmologist's office. So in the blue column, you'll see that that is the treated eye with gene therapy, and the red bars are the untreated eye. So any eye disease, you're fortunate because you have your own internal control, in a sense, since you have two eyes. So we have the treated eye and the untreated eye, and you can see these are the first 6 patients. So you see the first patient had a rather significant improvement in their visual acuity, 22 letters, or the equivalent of 4 lines on a Snellen chart. So they were able to see an additional 4 lines down on a Snellen chart. What's typically considered clinically significant is 10 letters to 15 letters. So they had a 21-letter improvement here, and alternatively, the untreated eye has continued to get worse. So it's deteriorated by 26 letters, or almost 5 lines. So that patient in one eye is nearly functionally blind, and in the treated eye is seeing much better. Similar story for patient 2, a significant improvement in the treated eye, plus 18, and the untreated eye is continuing to get worse. Patients 3, 5, and 2, so the middle clutch of patients, these patients came into the trial seeing at 20/20. So there wasn't much room for improvement, and in fact, you wouldn't expect much improvement because they're already seeing at essentially 20/20. In patients 5 and 2, it's starting to tail off. So they've lost -8 and -10 respectively, which speaks to the deterioration in the untreated eye. Patient 6 actually didn't get a full dose because the retina was stretched. This procedure requires a subretinal injection. So you have to go in with a needle, detach the retina, and put the vector in the subretinal space. It's very delicate tissue, and often in these patients, it's very fibrotic because it's advanced disease. And in this particular patient, number 6, Robert had difficulty, number 6 here, had difficulty detaching the retina, so he had to back out. So essentially what you've seen here is the course of the disease just continuing to get worse, which is what you would expect in both the treated and untreated eye, since the treated eye didn't get a full dose. Next slide. I mentioned the pipeline that we're developing. So beyond choroideremia, we have AAV8, so it's a different serotype we're using in a specific retinitis pigmentosa disease. So retinitis pigmentosa, very similar to choroideremia in the sense that it leads to photoreceptor death and blindness. There are multiple forms of retinitis pigmentosa, and we are looking at a very specific form. And we will be dosing patients in the UK in the first quarter of next year. We also have a macular dystrophy, which is, again, an inherited disease that causes a fluid buildup in the macula. And we will be dosing patients using adeno-associated virus in the fourth quarter of next year. So we have 2 new programs going into the clinic. And then, as I mentioned, we have a number of other programs that we're looking to advance into the clinic, again, all focused on using AAV, adeno-associated virus, and a specific gene to treat these various diseases. So open to any questions when we get to it.
Thank you very much. We'll now be moving on to Helix. We've got Tim Gideon and David Cavella from Helix, and they're going to be discussing their company. So over to you guys.
Great. So thank you very much, Hannah. So my name is Tim. I'm the Chief Executive of Helix, and this is David, who's our Chief Scientific Officer. And our startup or our activities are slightly different than the previous talks, and I think will fit quite nicely. So just a bit of what we're going to cover. So I'm going to explain a little bit about our model at Helix and a patient-driven model where we work directly with the patient groups and charities, and also explain how advanced data analytics, so machine learning, artificial intelligence, can be used to support drug repurposing and drug response prediction. So how a patient will best respond to a particular drug. Now, a bit of background. So we work from Cambridge University and bringing together heavy drug repurposing expertise and data analytics. So our CTO, the lecturer in molecular informatics at the university, and we are developing as a technology platform and a tech company. So we don't develop our own products, which is quite different than the standard biotech company. Now, the approaches we're using can be simplified and explained in two ways. So on one hand, we apply genomic matching, and on the other, literature and database mining to really predict drug repurposing, so how you can use an existing drug for a different disease and drug response. Those are a few examples of the patient groups and foundations we work with, and so there are a number of them. And I think what's really interesting is that we are now seeing that they play an enormous role within the drug discovery progression, and we really believe they should be also in the driver's seat, and they are the experts in their particular diseases. So we've set up a model where we work directly with them, which shown to be working extremely well, and we'll explain a little bit more about that. So we also developed tools for the patient charities, and that's one of them. It's called Omix, and it allows you to keep track of all the scientific literature around a particular disease, get explanations on the different terms, and that's an example for the CDKL5 forum, and they liked it so much that they've embedded it on their platform and website. And so that's quite a different way of working with the patient communities, which we think works quite well. Now, our model, we think, is really position ourselves as the honest broker between biotech companies and pharma companies, and then the patient charities and patient communities, and acknowledging that actually the patient communities are key opinion leaders and experts in their disease, particularly in rare diseases. So they can also provide access to patients, help generate key genomic data, support the development of animal models, cellular models, so that you can test the drugs. And so we really believe they have a key role to play there, and we've received a lot of traction for the model. Now, I'm going to hand over to David, who's going to explain more about repurposing and some of the projects we've been working on.
Thank you very much, Tim. Just a few words about the strategy of drug repurposing, which may be familiar to a certain extent with your audience, but some of these features may be less familiar. There are three main advantages to repurposing. It reduces the time, reduces the risk, and reduces the cost. The reduction in time mainly accrues during the drug discovery period, where you have about 2- to 3-fold quicker advancement of products through this phase, and then subsequent to that, entry into human trials. The reduction in risk is something that involves a reduction in drug development risk. So the probability of a drug entering into clinical trials finally being approved is increased by about 2.5-fold. And that's mainly a reflection of the reduction in safety risk, while the efficacy risk is mostly maintained. Put those two things together, you can calculate a reduction in cost of around 6-fold. So taking a metric of an R&D total cost of 1.8 billion, that can be reduced to 0.3 billion. And this is a major advantage for rare diseases, where the frequency of the disease can be a limiting factor on whether a drug can be developed for that particular condition. And we heard earlier from Glyn about the pricing difficulties that may accrue with NCE type approaches to rare diseases. Finally, of course, repurposing offers method of use patents. Now, in terms of rare diseases, repurposing has additional advantages because there is orphan marketing exclusivity, which applies in the major markets, 7 years in the US, 10 years in Europe, and also other rules and regulations apply in various other developed territories. Additionally, 80% of rare diseases have a genetic cause, many of them monogenic, and that means that using genomic techniques, we can apply mathematical algorithms to identify repurposing candidates. And we've done that in collaboration with a rare disease charity called APOD. This is an example of one of the charities we've worked with, and it focuses on childhood medulloblastoma. So medulloblastoma is a condition which affects more children than adults. In adults, it's an extremely rare condition, whereas in children, it's more common but still rare. That means that the medical burden is mainly borne by children, yet, as you all know, developing drugs and doing pediatric trials is a great challenge, and there's a significant medical deficit of products in this area. The approach we took with APOD was in 4 phases. Firstly, we applied clustering techniques to identify subtypes of the condition. Secondly, to each subtype, we applied algorithmic predictions based on transcriptomic matching. Thirdly, we refined those predictions using a proprietary repurposing database and literature mining techniques. And finally, and we are in this phase, we are validating these predictions using in vitro experiments, and we're doing that in conjunction with APOD itself. This slide here, very briefly, I won't dwell on it in too much detail, but it shows the clustering of 3 subtypes of medulloblastoma amongst the data set that we were able to access publicly. We then applied the repositioning algorithm to each of those subtypes separately, and I can describe the algorithm in this way. If you can imagine along a graph, the x-axis of the graph containing 20 to 30,000 expression profiles of various RNA, and on the y-axis you have the levels of expression of those genes, then you can imagine that in silhouette you have a mountain range profile of all those genes. What we're seeking to do is to inversely match the drug-induced change in expression level with the disease-induced change in expression level in a similar way to the reflection of a mountain range in a mountain lake. And we can apply this technology both for the identification of particular approved drugs that might be used for a rare disease, or we can do it in the inverse sense, where we apply it to an abandoned asset, say, from a pharmaceutical company, and identify which particular rare disease might be treatable with a particular drug. We validated the matching technology in the case of leukemia. In this situation, we predicted of the 30 top predictions, 24 of them were positively validated, 18 by reference to literature precedent, and of the other 12, 6 were new things which were identified and validated experimentally in vitro, the most potent of which had an LC50 in the nanomolar range with 40-fold selectivity over somatic cells. Typically, we take the predictions that have been developed, and this is what we did in the medulloblastoma situation, and reference them, comparing their occurrence using a proprietary database, drug repurposing database, which is a highly curated, large database of real-world literature in drug repurposing. which is categorized by mechanism, divided into different divisions of indication, and each individual reference categorized by level of evidence, whether it's clinical, in vivo, or in vitro pharmacological. And secondly, we use a text mining tool, which Tim has explained earlier in the presentation, called rare Omix, which includes processes involving Bayesian logic and natural language processing, and then a sequence of ontologies to find synonyms and so on. We could also use literature mining independently of genomics, and we have done this in the context of CDKL5 syndrome, supported by the Lulu Foundation, and identified a new drug, it's a new prediction of an old drug to be useful in this condition, and have found experimental evidence in a functional sense showing increases in expression of the relevant receptor. And then finally, we have deployed the gene matching technology in a different setting, and that is to predict the response of each individual patient depending upon their gene expression profile in the context of multiple myeloma. We've deployed this in 2 data sets and showed a significant difference between responders and non-responders, which we think can be used to enable us to reduce the level of non-responders in treatment of this condition by about 70%. The advantages of this, which we think can be applied to any drug and any disease, are obvious. We are now rolling it out into different conditions where we can gain access to the relevant data, and we also aim to apply it to multiple classifiers for more than one drug at a time. So just hand over to Tim to wrap up.
Yeah, so just to summarize then, I think there is really novel models to be established led by patient groups where you work directly with them, and I think we've explained a little bit about that. And then also the use of basically genomics and machine learning to help identify either the best rare disease for a drug or the best drug for a particular rare disease, and also expand these to individual drug response. So we'll be happy to take questions about this afterwards.
Thank you very much, Tim and David. We will now be moving on to our next speaker, who is Alistair McKinnon, Chief Medical Officer of Mereo Biopharma. So over to you, Alistair.
Thanks very much. Have I got control of the slide?
Yeah, let me just...
Okay, maybe just go on to the next one.
Okay.
Thanks. The next. So following last week, I'm pleased to say that I have to give the same disclaimer that Lynn gave. So Mereo is now a public company, so forward-looking statements to be treated with respect that's due. We'll move on. So we're just over a year old. We were founded in March of last year, and we're a specialty pharma company focusing on hospital specialty diseases and rare diseases. We raised a significant amount of capital, both about a year ago and again last week, to progress 3 mid-stage programs. All 3 of these came from one pharma company, from Novartis. The 3 founders had significant experience from doing VC deals of pharma spin-outs, and we felt that there was pressure within pharmaceutical companies to look at any way of fully investing in their pipelines, and that includes spinning things out. And we worked with Novartis for about a year to 18 months to identify 3 compounds that fitted our criteria, and one of these is BPS-804, which I'll talk mostly about today, which is an orphan disease, and the other 2 are in muscular specialty diseases. So I think our business model fits nicely on the back of Helix in that we feel that there's technology and assets sat within pharma that can be pushed forward more efficiently and hopefully more cost effectively outside of it. And our focus, as I said, will be on the orphan diseases going forward. Okay, next slide. So we're looking at osteogenesis imperfecta, or brittle bone disease. It's a rare disease with about 20,000 patients on each side of the Atlantic identified, and about the same number again. It's quite variable in how severe it is, but all patients suffer from recurrent fractures with minimal trauma, and that's how they usually present as quite young kids when they'll come into their A&E having had a fracture that doesn't make any sense. And there's obviously the concerns of the parents because there are questions around child abuse, but nowadays it's quite easy to get the genetic test to prove that it's OI. There are some very severe cases, and I'll come along to describe the different types, where kids are born with so many fractures, they're actually born with so many fractures that they don't really survive the first year of life. But those that do end up often quite short with significant scoliosis, progressive loss of hearing, problems with teeth, and in the end, they can end up with respiratory complications from that scoliosis. Most of these are linked to a mutation in type 1 collagen, and that's the first 4 types. There are now up to 16 types, I'll talk about 4 others, which are not collagen-based diseases, and our technology won't address those, but they're the minority of the patients. Okay, next slide. So this is the classification. There are 8 key forms. The first form, as I said, are collagen-based, mostly autosomal dominant, and type 2 is the most severe, where they have particularly abnormal collagen, and it's invariably fatal. Types 1, 3, and 4. Type 3 is the most severe, and type 1 the least severe. Type 1, the patients largely have just a reduced amount of relatively normal collagen, and type 3 and 4, they have mostly quantitative, but also qualitative dysfunction in their collagen. Okay, next slide, please. The current treatments, there's not much really out there. Fractures are managed largely in the way that most fractures would be managed by orthopedic surgeons, and sometimes prophylactic rodding is used in the long bones, but the rods themselves can cause quite a lot of problems. A lot of patients who experience a number of fractures early on often take to wheelchairs for the rest of their life, and bisphosphonates are used off-label, on and off. They're used variably in children and not so much at all in adults, and very little in the States. So it's really just symptomatic improvement, and there's not been much innovation in the field at all. Next slide. So our technology is a fully human antibody from Novartis, which is a sclerostin inhibitor. Sclerostin is a protein that's encoded on the phosphogene and is secreted by osteocytes, or mature bone cells. And this inhibits the osteoblasts, reducing bone formation via LRP6 and the Wnt pathway. And what's quite nice about using an anti-sclerostin is there are human models of this. So there's 2 very rare diseases, Van Butten's and sclerosis, where patients genetically produce no or almost no sclerostin, and they have very, very strong, thick bones. Most of the patients are in South Africa, and they're known to have barely suffer fractures and walk away from car crashes and things like this. So it's quite nice that we can see, from a safety point of view, that inhibiting sclerostin to a significant level doesn't cause problems over the long term. The data we have from Novartis is in nearly 100 patients across 3 diseases, hyperphosphatasia, OI, and postmenopausal osteoporosis. And they clearly show across all of them, but particularly in the OI patients. But this is a powerful builder of bone. It also reduces bone resorption and shows statistical significance on all the commonly used blood biomarkers and anabolic effects on DEXA BMD. Next slide. So, over the year, since we started, there was just 3 of us here about a year ago. One of the key things we thought we needed was someone who is an expert in the development of orphan diseases to lead this program, and indeed other orphan compounds that come into the company. And we were able to hire Dr. Tony Hall from BioMarin, who's leading the program. He's successfully got orphan drug status for us in the US and secured it subject to EU recommendation further in May this year. Along with what a lot of the other speakers have said, we've engaged with the community, and there's been very little engagement with the OI community from industry over the years, and we've rapidly got involved with all of them. There are 2 main groups globally, the OIS in the States and the BBS over here, and the adoption of the technology of the company by the patient groups has been fantastic. So we can echo exactly what Lynn was saying earlier. We're also working with a group in Oxford who's called the RUDY study, who've been looking at rare diseases in bone and connective tissue and creating web-based systems for real-world data. So we're adapting their technology to have patient-reported outcomes and patient-reported fractures as part of our next study. Given that this is obviously a disease that patients live with for quite a long time. Then next slide, please. I think one of the things I'd like to mention about rare diseases is the use of novel endpoints. And in bone disease, BMD by DEXA has been used as the gold standard for years, but it's often been criticised as not being particularly predictive of fracture risk. And over the last 15 years, a new technique called HRPQCT, or High Resolution Peripheral CT, has been developed. And this gives us 17 variables across bone and allows a technique called finite element analysis to be used to measure bone strength and predict fractures. And finite element analysis comes from civil engineering technique of the strength of hollow or structures. And in the chart below, you can see what an HRP-QCT slide shows in OI patients. So the normals on the left, the less severe patients is at type 1, the second one in. Type 4 is the next least, which shows almost no trabecular bone, and type 3 on the right is the most severe form, showing cortical and trabecular disease. So this is the technique that we're proposing to use. It's getting more and more, it's getting a lot more traction because in the osteoporosis community, it's explained why women with a normal aerial BMD on DEXA can have fractures. And when you look at their HRP-QCT, they've got an aerial normal BMD, an abnormal volumetric or quantitative BMD. The next slide. Thanks. And so finite element analysis showed that when you looked at osteoporotic women, 40 of them with and 40 without fracture, it clearly defined the patients who are likely to fracture. And in a study of 80 OI patients, it also showed the effect of Forteo, which we believe to be an inferior way of building bone, but did show a statistically significant increase in the bone strength and reduction, importantly, in fracture risk of patients with OI, and this was across OI 1, 3, and 4. Next slide, please. So the study we're planning to do is also quite a novel way of doing a rapid development program. So we'll be using HRP-QCT to select a dose in a seamless phase 2-3 study, and we will look at fracture, time to first fracture as being the final endpoint, but following an interim analysis with HRP-QCT and finite element to try and demonstrate patients who are at most risk of fracture. The next slide. And so our focus going forward is to continue on rare diseases. We've got an active pipeline from the large pharma companies. We're looking for similar early proof of concept data, and we've found a number of pharma companies that have been using orphan diseases as a proof of mechanism for their larger indications, but where they wouldn't take it forward in orphan. And when they've done the phase 2A and the larger indication, it hasn't shown the data that they would need to push forward, but there's still the potential in the orphan disease, and that's a key space for us to look in. And that's Mario today. Thank you.
Great. Thanks very much, Alistair. I will now be moving on to see if there's any questions from everyone. So I'm just going to unmute everyone. If you don't have any questions to ask, please remute yourself. It might be a bit loud. So I'm going to open up the floor now for any questions. So please talk if you have any questions. Okay, so I've got some questions to read out here, if no one has any, read out loud. The first one is to Glenn. How sustainable is current orphan drug pricing? And other people can also pitch in later. So Glenn?
Yeah, that's a really interesting question, because it's going to drive a lot of the investor interest in the sector. And I think orphan drug pricing is quite sticky. So I think long term we've got to plan for a lower level of orphan drug pricing, but there'll be large variability, and it really depends, I think, increasingly on the benefit that you give. So I think there have been some cases of severe pressure on some recent orphan drugs, but then if you look, the actual benefit that they give is quite minor. But on the other hand, in some quite large indications, if the benefit to both the patient and society is huge, then you can get the pricing. So I think the days of if it's an orphan indication, you can get a great price if you get approval. That's probably going to be less true in the future, and that you'll have to show more of an economic case, is my view. I think the overall macro thing just to consider is, you know, each of these diseases might be rare in its own right, but then if you put them together in aggregate, it comes to quite a lot. And I've got no validation for it, but I did sit at a conference in the US a little while ago where someone said 1 in 7 Americans has a rare disease, and that's a bit of a worry if we had treatments for all those people in terms of the economics of it.
Thanks, Ben. Does anyone else have any thoughts on that?
Yes, so we'd like to add that I guess one way to help patients without having the challenges of all this high pricing is repurposing. And there are people in the US, and a particular chap called Bruce Bloom, who is running Cures Within Reach and started Cure Accelerator, and they help basically fund pivotal clinical trials for repurposing candidates, and then once they've shown to be safe and effective, then go towards an off-label route of using the medicines for those rare disease patient communities, which is not ideal. It's a gray area, but it's a way to go quicker to patients and also avoid this high pricing challenge. And David here has just written a book on off-label use of medicine, and it's very challenging but interesting area.
Thanks, both. Does anyone else have any thoughts? David or Alistair at all?
This is David. What I would say in gene therapy, of course, the big question is how durable is it? So can indeed you administer the gene and it'll continue to express and treat the disease over the long term. And of course, the only way we'll know that is by doing the appropriate studies and then getting into real world experience. So in some ways, the sustainability will depend on how efficacious over the long term the gene therapies pan out to be. At least for us, the early indications are good, that we will continue to have a durable expression, but again, that has to be borne out in the real world experience. I guess the only other point I'd make on the sustainability is, I think to the extent that we as developers can bring forward a case where we show that we're eliminating some cost somewhere in the system, not only adding cost, but we're eliminating cost, to the extent that we can develop those arguments, I think we have a much better chance of realizing these larger price tags.
Thanks, David. I do have one specific question here for Alistair as well. So, what other compounds have been in clinical trials for osteogenesis imperfecta, and how did they perform?
Yeah, the bisphosphonates have been in some trials, but there's not been anything done over a long enough time to really show a difference, and are also less powerful compounds. And I showed some data from the HRPQCT FEA study on Forteo, and that was probably the biggest study in OI patients, 79. It was a blinded study over 18 months. And that's the primary endpoint of that was aerial BMD, and its secondary endpoint, which was the HRPQCT, but which they hit. But people really probably want to see fracture or a reliable way of predicting fractures, which is why we're doing the FEA. And that goes to the comments that David and Glenn have just said about pricing, because people will want to see that we're reducing the cost of the healthcare liability coming from these patients. They're fracturing less, they're having less operations, they're not needing rodding or physio. So there's nothing that's been successful, but there has been one proper trial. It still wasn't done by industry. It was an academic trial, but it was well done.
Thank you. Probably time for one more question, and this is originally for Glenn. Is the UK a good place to run clinical trials for orphan drugs?
It's Glenn here. So I've got two conflicting bits of data. The first is, for any translational medicine, I think the UK still remains to be a fantastic place to run trials, because you've got really expert physicians in close contact with the scientists who understand the biomarkers, and so that aspect works really well. But the speed at which sites get up and running, the rate at which they recruit patients is just awful. And, you know, sadly, in our phase 2 Duchenne trial, which we expected to recruit the majority of the boys in the UK, despite getting regulatory approval in the UK and having loads of parents and boys ready to go, it looks like most of the patients will be recruited in the US, where actually the whole process in the US started about 6 months later. And it's not due to enthusiasm or anything, it's just that the health service is so lean that they just don't have the capacity to do stuff, and it's just awful, just awful.
Excellent. Does anyone else have any thoughts on that? If not, I think that's probably all we have time for in terms of questions. I just wanted to quickly highlight our next webinar, which is on the 12th of July, and it's on raising capital in current market while retaining control. And this is a Horizon Discovery webinar, so Darren Gisley will be on that, and we'll have a couple of other speakers too. The webinar itself will be distributed to everyone who signed up, and it will also be available on our YouTube channel, so keep a lookout for that. I just want to highlight a few of our upcoming events. If anyone doesn't know already, the CEO Investor Forum is next week, in Hertfordshire. We've got our summer reception on 7th of July, which is taking place in a really nice rooftop terrace overlooking the Thames. Women in Biotech event on the 14th of September, a breakfast in Stevenage in September, and I just want to highlight the Bioscience Forum is on the 20th of October, and it is now open for registration for early bird rates, so do put that in your diary. Finally, I just want to say a big thank you to the 4 companies that took part: Summit, Nightstar, Helix, and Mereo Biopharma. Thank you very much for taking part, and I hope everyone enjoyed it. So on that note, I just want to say thank you for listening, and I'll bring the webinar to a close now. Thank you.
Thank you.
Thank you.
Thank you.